So let's continue with our discussion of Hardy-Weinberg condition. So remember, these are conditions that have to be met if a population is not evolving. If any of these five conditions are violated, then that means that this condition is causing a change in allele frequencies over time. So we've already talked about no natural selection, no mutation, no migration. Our next one is no genetic drift. So genetic drift is a change in allele frequencies due to chance. So we've seen this due to chance thing before when we did our genetics unit and we were looking at, we expect one quarter of our population to have this phenotype, but if you look at the actual observed numbers, it's slightly more, slightly less. So it's the same thing in any population. Just by chance, less individuals might be born that are heterozygous or homozygous than you would expect, simply by chance. So this is also evolution, and students often have trouble understanding that you can have evolution without natural selection. So this is one of those cases where a chance event, just by chance, none of your four offspring happen to have the recessive phenotype. Because remember, each event, each birth is an independent one. So they don't all come out, all your children don't come out in a 3-to-1 ratio because they're supposed to. Each one has a 1/4 chance of having a recessive trait. So this is simply changes due to chance. The other thing that this is called sometimes is large population size or infinitely large population size. And the reason that it's called this is that large population sizes, because there's such a big number, are going to be less likely to be affected by chance events. Doesn't mean they can't happen. They absolutely can, but the impact of those events is going to be smaller. So it's kind of the idea of thinking, if I flip a coin 10 times, I expect 5 out of 5, but I don't always get it. Sometimes I get 4 and 6, which is kind of a big difference when you only have 10 individuals. But if you flip a coin 1,000 times and you get 449 and 551, that's a really small difference compared to the number of individuals. And so that's why this can be called population size. So you want if you are meeting Hardy-Weinberg conditions, you want a large population size. And there's no genetic drift. So any chance events that might occur are not actually going to affect the population because it's such a large size. Small populations are more strongly affected by chance changes. If you have a small number of individuals in a population, and for some reason unrelated to natural selection, your only individual that happens to be homozygous recessive dies, then you've just lost some of your alleles. So no genetic drift is pretty important. This is really an unpredictable event from generation to generation. Not all individuals are going to leave offspring, and it has nothing to do to natural selection. Sometimes the flower doesn't get pollinated, or sometimes a goat eats the flower before it can be pollinated. It has nothing to do with whether it was the right color or the right shape. Just maybe the bee got tired, or maybe the goat got hungry and happened to miss the flower or eat the flower. So this diagram sort of shows how this fluctuation can come about. So we have a population on the left that has pink, yellow, and white flowers. And if you look, you can sort of see their relative frequencies. Red and pink are roughly equal, and then there's the one little lonely white flower there. Um, but let's say only those ones that are highlighted in yellow, for whatever reason, leave offspring. Maybe that goat walked down the middle of the path or walked down the middle of a meadow and ate everything down the middle and then left stage right. You know, maybe that's the path that he took and he didn't care what he was eating on the way out. And so the only flowers that were left are these ones highlighted in yellow. So just by chance, you can now see in our center image that there are far more white flowers and less pink and red. So we've had this change that has nothing to do really with the color of the flower. Let's say now two goats come through and they eat everything that's in the middle of the meadow and they miss those two red flowers on the outside. They're not picking colors, they're just picking flowers. So if only those two flowers survive, the next generation is going to be all red. So if only those red plants leave offspring, now our entire population is red, simply by chance. Not because red is better than the others, but simply by chance. Of course, you could make the argument that maybe the goat was selecting them or whatever, but what I'm saying here is that simply by chance, there was no reason that those red flowers should have been left compared to any other flower. So this can happen due to a natural disaster, so a forest fire or a flood, where individuals are not selected based on any trait. They just happen to die because they're there, or just for one reason or another they don't leave offspring. So it really has nothing to do with the trait that they have, but those allele frequencies can change in the population. So down on the bottom, we actually have a new notation here. We have P, the frequency of the red allele equals 0.7, and Q, the frequency of the white allele equals 3. And you can see that it changes across these three generations. P continues to increase while Q continues to decrease. I just want you to keep those in mind because we're going to talk about what those mean and how you calculate that in a few minutes. There are a couple of ways that genetic drift, specific types of genetic drift, and so let's talk about those. One is the founder effect. So a founder is somebody that starts a new population, or a group of somebodies that start a new population. So the founder effect is when a few individuals become separated from their parent population and they establish their own new population. So just by chance, because genetic drift is a chance event, just by chance, that new population has different allele frequencies than the parent population. They didn't leave because they had particular alleles. They left and happened to take whatever alleles they had with them. So this has nothing to do with whether or not a trait is advantageous or not. This example here, this is the island of Tristan da Cunha. It's an island you can see there in the middle of the Atlantic Ocean, the southern Atlantic Ocean, between South America and Africa. And this is kind of interesting. In the 1800s, 1814, 15 British colonists founded this island. So very small population from the original British parent population founded this island. And one of those individuals happened to have something called retinitis pigmentosa. Now, this is a type of progressive blindness. So this individual would have had this disorder and founded this new population. And now, a few years ago, someone studied this island and sort of took a survey of the descendants from the original 15 colonists. And they found that out of 240 descendants, 4 individuals had this form of progressive blindness. And that might not sound high to you. It's a very rare disorder, but it is actually 10 times higher than the frequency in the British population. So just by chance, this population happens to have a high frequency of this allele for retinitis pigmentosa. has nothing to do with why they founded the island. Just by chance someone happened to have that allele when they got there, and now the descendant population has a very different frequency than the British descendant population. Second type of genetic drift is something called the bottleneck effect. So the bottleneck effect is important for a couple of reasons. The bottleneck effect is when there is a sudden change in the environment. So this sudden change in the environment may reduce the population size. So this can be all sorts of things, and the rate of the population size really, the rate of the decrease in population size is what's important. It's a quick decrease in a short amount of time. So in this situation, with the bottleneck effect, by chance alone and that quick reduction in population size, allele frequencies change. So individuals are not selected for any particular allele, but a whole bunch of them die really quickly. If you look at this bottle, that's why they call it the bottleneck effect. Look at this bottle on the right. You have this wine-shaped bottle full of alleles. You have an event where only some individuals survive in a short period of time. And if you look, there are red, yellow, and blue alleles in there. So this picture is demonstrating that if only a few individuals survive, you can have a change in allele frequencies. And you can see that in our surviving population, that yellow allele is not even present anymore. And this is simply by chance. The individuals didn't die because they had the yellow allele. They died due to some sort of environmental change or some sort of environmental impact that killed a large quantity of population very quickly. So the example for this is the Florida panther. These guys were hunted and killed over a relatively short period of time because they were seen as nuisance animals. And so in a short amount of time, the genetic diversity of this population was drastically reduced. The population number was drastically reduced, and the variation present in that population was also reduced. The bottleneck effect can be really important in the genetic variation in a population. So not only are the allele frequencies different in the next generation, but because you have a small population size now, genetic drift or just random fluctuation in allele frequencies can cause even more problems. So let's say that one individual that is white over here, for whatever reason, doesn't have any offspring. Now you have no white alleles either, just by chance. Excuse me. And so you can see that not only do we have a small population size, but we have low genetic variation, which is going to limit the way that they can respond to future selection pressures. The other thing that happens here is going to be the violation of our last Hardy-Weinberg condition, and that is non-random mating. So when you have a really small population size, those individuals are forced to interbreed, and you get sort of related organisms or more closely related than normal that are forced to interbreed because there are no other options. And this can also affect the genetic diversity and the survivability of that population. So another example of this that I think is kind of cool that our book uses is this Illinois prairie chicken. So this is the chicken there on the right. He's a little bit funny looking, but he's got some really pretty coloring. So this is the historic range for the Illinois prairie chicken, and this is when Illinois was covered in prairie, when most of the state was prairie grassland. They had huge population numbers. Several thousand of these individuals living in Illinois in their natural range, which was basically the whole state in the natural prairie. So before the 1960s, there were estimated to be up to 25,000 individuals in Illinois, and then as the prairie got turned over for development and for farmland, by the 1990s there were less than 50 Illinois prairie chickens left. So in a span of, say, 30 to 50 years, the population size went from many thousands to 50. So there's a crazy decrease in population size over a short period of time. So the problems with this, one of the main problems is low genetic variation. And then of course you also have inbreeding. So that population is small. There's not a lot of options for breeding. And so when you have a situation like that, you sort of magnify this low genetic variation problem. And so they had a low hatch rate, which means not only were there not a lot of individuals to lay eggs, but then a high number of those eggs were not even hatching. So you can see on the far right there, it shows the number of alleles for the average number of alleles per gene decreased from 5 down to 3. And the percentage of eggs hatched went from 93 down to less than 50%. So the survivability rate and the genetic variation was vastly different after this bottleneck effect. On a positive note, the prairie chickens in Kansas and Nebraska did not suffer such a terrible fate. They did not experience this sort of bottleneck effect. And so somebody, I don't know, Department of Natural Resources, somebody established a program to actually bring in prairie chickens from Kansas and Nebraska to increase the population size here and to increase the genetic diversity. And they were actually able to restore it back to closer, at least the genetic diversity and hatch rate has been able to increase. You can see now, though, there's very little prairie for this prairie chicken to live in. So that's still a problem, but the population was able to recover somewhat. Our last Hardy-Weinberg condition is non-random mating or inbreeding. So non-random mating is when individuals either are forced to choose an individual with a specific trait or choose an individual with a specific trait. The other part of this is inbreeding. So this is breeding with close relatives. So the condition is that there is neither selection for a specific trait or inbreeding in a population. So there's a lot of things that are covered by this. And what random mating means is that any genotype has an equal chance of mating with any other genotype. So really that means a random bunny rabbit will mate with any other random bunny rabbit for no particular reason. They're not choosing any specific traits. They are unrelated, at least not closely related. And so with respect to the alleles and the genotype, everything is random. This is violated if individuals tend to mate within a subset of the population. So when this happens, you don't really get a random mix of gametes. You get a mix of a small set of gametes. So that's going to have an effect on your allele frequencies and your variation in the population. This also means no sexual selection. So sexual selection is when individuals are chosen based on a particular trait. So think of birds with really showy feathers, or deer or moose with really big antlers, a lion with a big mane. These are traits that females are looking for and choosing in their mate. So this also means that the specific trait that you're looking at is not being chosen as part of their criteria for the mate. Again, as I was saying with bottleneck effect, sometimes the individuals have no choice. If there's only a small population left, you are forced to inbreed. But this can also occur in large populations. If for some reason individuals are mating with close relatives, this is a problem, or if they're choosing specific traits, you get non-random mating.